Assalamualaikum and a very good day. My name is Siti Nur Afiqah Binti Mahazan, a final year student for the Chemistry Forensic Analysis program under the supervision of Dr. Wan Nazihawan Ibrahim. I would like to present on my final year project based on Mode 1 entitled Pre-concentration of organochlorine pesticides using microsolid phase extraction based on bio-sorbent alginate-silica beads derived from oil palm frond. For the background and problem statement of this study, oil palm frond, or known as OPF, is one of the primary biomass products of oil palm plantations. It is suitable to be used as a raw material for some value-added products as it is abundant, readily available, and inexpensive. OPF also contains silicous material which can be extracted for further research, and the production of OPF silica is an alternative to reduce the production of commercial silica. Alginate contains multiple chemical and physical cross-linking, functionalization, and reactive sites due to its lots of hydroxyl and carboxyl groups. By encapsulating alginate with silica, it would limit the quantity of alginate hydrophilic molecules, influencing their breakdown kinetics and improve the integrity and mechanical qualities. For the organochlorine pesticides, or known as OCPs, it have been widely used in agricultural pest control, especially in paddy plantation. It is present at extremely low concentrations, which makes them difficult to be detected. And to detect them, it requires efficient and selective methods. For the conventional extraction methods, it usually utilized through liquid extraction and solid phase extraction, but it does not fulfill the concept of green analytical chemistry as it requires large amounts of organic solvents and generate hazardous waste to the environment. Thus, for this study, micro-SPE technique has been utilized as this technique is fast and uniform solvent-analyte interactions can be made and require low consumption of organic solvents. The significance of this study is to enhance the use of agricultural waste to produce more eco-friendly solvent instead of producing commercial silica. Also, it is to reduce the cost of analysis and time by developing and optimizing the extraction parameters to be used to extract organochlorine pesticides using a simpler and less hassle sample preparation method. For the first objective of this study, it is to examine the chemical and physical properties of alginate-silica beads prepared via the encapsulation method prior to the characterization using ATR-FTIR and FSM analysis. For the second objective of this study, it is to evaluate the performance of the prepared alginate-silica beads in microsolid phase extraction to extract organochlorine pesticides in the paddy water sample and optimize the parameters including extraction time, desorption solvent, desorption time, and mass of solvent. For the literature review part, oil palm frond consists of three main fibers, which are cellulose, hemicellulose, and lignin. The OPF contains more cellulose than other waste portions of oil palm trees, and the accumulating quantity of OPFs could serve as an inexpensive source of cellulose that could be used for a variety of applications. In comparison to other biomaterials, this organic polymer has superior properties such as high water absorption capacity and large surface areas. The abundance of this biomass made it a viable alternative as a silica precursor for the synthesis of silica nanomaterial from agricultural waste. Through the incorporation of silica-based domains, the synthesis of alginate-based composite beads is an attractive option. Due to its excellent physical and chemical stability as well as its acceptable biocompatibility, silica has been utilized. In addition, silica does not significantly alter the optical or magnetic properties of the components it is combined with. Lastly, the hydroxyl groups on the surface of silica particulates make chemical surface modifications, and the incorporation of functional groups simple. By controlling the pore volume, pore size and tortuosity, and the surface chemistry of silica particles allows for the customization of their release profiles for control release applications. Figure 2.2 shows the synthesis mechanism of alginate-silica composite adsorbate. Organochlorine pesticides is a typical persistent organic pollutant whose toxicity and environmental impact have been a global concern. Despite the Stockholm Convention's prohibitions on the use of these pesticides, some developing nations continue to use these inexpensive and effective pollutants for agricultural purposes. To ensure environmental safety and human health, it is essential to develop a simple, rapid and accurate detection method for organochlorine pesticides in the environment. Table 1 shown the concentration of aldrin, dieldrin and heptachlor in water based on previous study. For sample preparation part, liquid extraction and solid phase extraction are conventional sample preparation methods that are frequently used. However, these methods consume large volumes of hazardous organic solvents, produce significant amounts of waste, and deem not safe for humans and the environment. Thus, for this study, microsolid phase extraction was proposed as a replacement for conventional multi-step SPE due to its reduction in the amount of solvent, desorption solvent, and sample volume. It is also suitable for processing relatively complex matrices and provide lower cost of analysis. Table 2 shows the analytical performance and extraction conditions comparison of the developed procedure with other recently published works. For the methodology part, the steps begin with the preparation of OPF-silica powder, which was prepared by the postgraduate student before preparing the alginate-silica composite bits manually in the laboratory. After the final product was obtained, characterization of the composite bits was done based on ATR-FTIR, FSM, and ADX analysis. Moving on to the next part, the evaluation based on the performance of alginate-silica composite bits in paddy water sample was done through optimization parameter based on extraction time, desorption solvent, desorption time, and mass absorbent. Then, preparation of standard and sample solution was done, followed by the preparation of microsolid phase extraction before proceeding with method validation part. Based on the observation from digital photograph of OPF-silica powder, pure alginate bits, and alginate-silica bits, OPF-silica powder can be seen to be a white, fine powder with spherical and super smooth particles. Pure alginate bits is brownish in color with unsymmetrical round shape and have hydrogel-like texture. On the other hand, alginate-silica bits is yellowish in color with unsymmetrical round shapes and have hydrogel-like texture but more firm than pure alginate bits. Based on the ATR-FTIR analysis, the FTIR spectra of OPF-silica powder show the existence of a broadband around 3,407 per centimeter, which shows the OH stretching vibration of the silanol or the absorbed water molecules on the silica surface. The strong band at 1,079 to 805 per centimeter corresponds to the asymmetric vibration of the siloxane bond, which forms the silica matrix backbone. The FTIR spectra of alginate-silica bits, on the other hand, demonstrate that the absorption peaks at 3,326 per centimeter corresponds to the OH absorption band, which is due to the stretching vibration of residual OH groups or water absorption. The absorption maxima at 1,029 and 824 per centimeter correspond to symmetric and asymmetric stretching vibrations of siloxane bonds, respectively. The bands at 1,598 and 1,419 per centimeter in the spectrum of the alginate-silica bits may be assigned to asymmetric and symmetric carboxylate deformation, respectively, which confirms the interaction of carboxylate groups with silanol produced from oil palm fronds. For FSM analysis, a clear comparison can be seen between pure alginate bits and alginate-silica bits, where the surface of pure alginate bits can be seen to be rough and lacuna with numerous creases. The alginate-silica composite bits also have a rough surface, but this is due to the cavity generated during the cross-linking process. It can be concluded that both pure alginate bits and alginate-silica bits have spherical shape, and this can be concluded that the similar structure of pure alginate bits and alginate-silica bits demonstrates the homogeneous distribution of silica in the alginate matrix. Based on the EDX analysis, it was revealed that pure alginate bits consisted primarily of oxygen, calcium, and a minor amount of sodium. The most significant difference is the presence of silicon in alginate-silica bits, followed by oxygen, calcium, and sodium. Due to the prevalence of silicon in the composite bits, this outcome was logical, as the results are consistent with those previously obtained in the FTIR section. For the optimization of micro-SPE procedure, 4 parameters were used, which are extraction time, desorption solvent, desorption time, and mass absorbent. It is to evaluate the efficiency and sensitivity of the extraction using distilled water. The parameters were carried out using one variable at a time, or known as OVAC, while other parameters were kept at constant. For the effect of extraction time, the impact of contact time between adsorbent and target analytes is depicted in the chart. It was discovered that the adsorption capacity of OCPs was fast between 2.5 to 15 minutes. The highest extraction efficiency was obtained after 15 minutes of extraction. For the effect of desorption solvent, N-hexane performed better than the other solvents regarding desorption. N-hexane is a non-polar with polarity index of 0.1, provide a more favorable performance in dissolving the target analytes due to the hydrophobicity properties of the OCPs. For the effect of desorption time, as a result of the reabsorption of the target analytes, the peak area of each target OCP steadily decreased after a 7-minute desorption time. Eventually, the optimal desorption time for further analysis was determined to be at 7 minutes. For the effect of mass absorbent, the trend of the graph can be seen to be decreasing after 50 mg absorbent mass was used. Since the peak area for the analytes was at its maximum of 50 mg, the sorbent mass of 50 mg was used for the following analysis. Based on table 4.2, it shows that the optimal values of the extraction parameters for micro-SPE were optimized, which is to be used for method validation part using paddy water sample. For the method validation part, prior to sample analysis, the linearity, limit of detection LOD, limit of quantification LOQ, precision percent RSD, accuracy percent recovery, and enhancement factor EF of the extraction method were evaluated. Based on table 4.3.1, it shows the performance of the proposed method under optimized conditions, where it can be seen that heptachloralgine and dialgine produce good regression coefficient at 0.9796, 0.9908, and 0.9935 respectively. On the other hand, Table 4.3.2 shows the method precision for intra and interday RSD, percent recovery percent ER, and enrichment factor EF, where the lowest and highest concentration of each analytes did not exceed 10%, and the percent recovery for lowest and highest concentration of the analytes used was in the range between 82 to 98%. Figure 4.3.3 shows the report chromatogram of water sample at unspiked blind condition, spike at lowest concentration, and spike at higher concentration. In conclusion, the physical and chemical properties of alginate-silica beads prepared via the encapsulation method can be examined prior to the characterization using ATR-FTIR analysis, PSM analysis, and EDX analysis. The performance of the prepared alginate-silica beads in microsolid phase extraction for the extraction of organochlorine pesticide in paddy water can be evaluated based on the optimum values obtained from the optimization parameter, which required 15 minutes of extraction time, and N-hexane as desorption solvent, 7 minutes of desorption time, and 15 mg of sorbent. For recommendation, future environmental applications of alginate-silica beads require further research on the mechanisms involved in the extraction of organochlorine pesticides for different water samples. That is all from me. Thank you for your time and cooperation.